Okay, so for this video, we are talking plasma membranes, cell membranes. We're talking structure and movement of molecules across that membrane. All right, so the key knowledge that we are focusing on for this section, the fluid mosaic model of the structure of the plasma membrane and the movement of hydrophilic and hydrophobic substances across it based on their size and polarity. So fluid mosaic model being the bits of the membrane and the role that each of them play. The movement of hydrophilic and hydrophobic substances, so two really key words that have cropped up again and again in the course that you need to be comfortable using and remembering when they apply. As well as the role of different organelles, including ribosomes, endoplasmic reticulum, Golgi, and associated vesicles in the export of a protein product from the cell through exocytosis. So this is all talking, you could shorten this up to everything in the cell that's involved in the creation and export of a protein from the cell through one specific method of transport across membrane being exocytosis. And then by comparison, endocytosis, so cellular engulfment of material by endocytosis. So these three dot points kind of gel together as the structure of plasma membranes and how we move substances across them, including the bigger, bulkier molecules through exo and endocytosis. Okay. Looking at the structure first, and I'll try to be nice and descriptive with the language, so if you are just listening to this video, you don't necessarily need to be looking at the diagram in order to understand. But we have a picture of what represents the fluid mosaic model of the cell membrane. So we've got not only the drawing of the phospholipids and their arrangement in that bilayer, how we've got the hydrophilic heads, the circular part, pointing out, and the hydrophobic tails pointing in towards each other to form that sandwich of the membrane. Not only the phospholipids, but we've got all those other little bits and pieces that go together to create that mosaic model of the membrane. So having a look at a few of those, what have we got labelled? We've got carbohydrate chains, and we can see those. Well, you could recognise that they're carbohydrate chains without the label anyway, because they're made up of those little hexagons that we come to recognise as a representation of a carbohydrate. And we can see those attached to two different structures. So if it's a carbohydrate chain that is attached to a phospholipid, we call it a glycolipid. So the glyco for the sugar part and the lipid for the lipid that it's sitting on. We can also have glycoproteins, which is a carbohydrate chain attached to a membrane, a integral protein in the membrane, a protein that's sitting within the phospholipids of the membrane. So they're usually just one that you would have to recognize the shape of and be able to name. The lipid and protein part just comes from whatever the carbohydrate chain is attached to. And the role that these things play is usually something to do with cell identification. It might be a marker, a self marker, or a different kind of antigen used to determine what kind of cell this is in the body. You can get these particular molecules that play a role in adhesion. So two cells sticking together. If you've got, say, muscle cells that are forming part of muscle tissue, you want them to be able to hold together. So it might be playing a role in adhesion, or it could be a part of receptor formation. So they're the roles that you would recognize those things doing. The bigger ones that we see here, and I'm pointing at two in particular, these are transmembrane proteins. So that's a word that should be familiar and sounds confusing, but it's really not. It just means that it's touching both sides of the membrane. So you can see this transmembrane protein poking out on either side. And we've got two distinct shapes. We've got one that you can see it's been cut in half, but you can see there's a clear tunnel through the middle of it. And you can see the other is just a solid protein. They would play different roles. So I would expect this one without the tunnel through the middle of it. This could be part of a receptor for, say, a hydrophilic hormone, where we need it to be able to respond to a change on the external surface, and its conformation would change. It will be able to pass that signal onto the inside surface. So remember that the hydrophilic hormone doesn't actually enter the cell. There's no way for the hormone to travel through this protein. It is a solid protein that doesn't allow transport, but because it's touching either side, it's able to pass a message from the outside to the inside. Whereas this structure would more likely be a protein channel, something that a molecule can pass through to gain entry to the cell. And you can see because it forms that tunnel shape. The one big thing that is missing from this diagram is something called cholesterol. So we should have another little structure that we would draw sort of as a irregular shape that's embedded inside the hydrophobic part of this membrane, and it could be labeled cholesterol. Cholesterol has that role that we need to remember in the plasma membrane of maintaining stability. So it's got a role at either end of the temperature spectrum. Cholesterol functions to keep the membrane together, so it's keeping it stable. At high temperatures, the membrane doesn't disband from each other and fall apart because of cholesterol playing its role in the structure of the membrane. So we've got cholesterol maintaining stability in high temperatures while also maintaining fluidity, so allowing the membrane to stay fluid and flexible in lower temperatures, preventing the membrane from freezing. So that's a look at the structure of the membrane. Two kind of questions you could be asked about membrane structure. You might be asked something about the fluid mosaic model, about a particular component. So you do need to know the role of these things that they play, what kind of job they have in the membrane structure. And the other type of question is about specifically the formation of the phospholipid bilayer. So that's not taking into account all of the elements of the fluid mosaic model. It's not talking about all the integral proteins, transmembrane proteins. If you're asked about the arrangement of lipids in the phospholipid bilayer, it's only asking you to draw, that's a very common question to ask, to draw the phospholipids in their formation. So with the hydrophobic tails pointing in, and the hydrophilic heads on the outside, and they sandwich together in that way. All right, so that is plasma membrane structure. Looking at lipids themselves, just move me down a little bit, as a macromolecule, things that we need to know and associate with lipids. So that's that phospholipid bilayer structure that I was just talking about. That's what you would be expected to recall, that is a single phospholipid with its glycerol head, phosphate head, and fatty acid tails, two fatty acid tails. And that's the chemical structure that it would take on. So if you were shown that, you would recognize it as being a lipid. You would never be expected to draw that chemical structure. Lipids themselves normally consist of, this would be a triglyceride, glycerol head and fatty acid tails. So this would be the glycerol head part, and there would normally be 3 of these carbon chains making up the fatty acid tails. In this version, instead of the extra fatty acid tail, we have a phosphate group attached to it. And that's what makes up the phospholipid, the phosphate head of the phospholipid. So that's this part here when we're drawing it as a diagram. But you should be able to recognize either structure as being a phospholipid. You would never have to draw the chemical structure, but definitely be able to draw this more basic version. types of lipids that we can encounter or other words that it might be given. Fats, oils, steroids is a common word to use to describe a lipid molecule, and phospholipids we're very familiar with. Okay, a couple of exam questions that you can have a go at. So pause the video here, actually have a go at answering the questions, and I'll get straight into the answer after a quick pause. Okay, so hopefully you've had a read and we can see the diagram shows a portion of the plasma membrane. What's been labelled here? Hopefully it jumped out at you that this is a carbohydrate hexagon that it's labelled, and that carbohydrate is attached to a lipid. So you would be labelling this as a glycolipid. It's not cholesterol because it's not embedded within the hydrophobic area. It's not a phospholipid. It would have to, the X label would have to be around the actual phospholipid. And a globular protein. Don't let words like this throw you off. You know what a protein is. Globular just means that, well, it would be this part here. So globular protein is a short, fat, folded up version of a protein, not a elongated structural protein. Okay, so hopefully we went with the A on that one. And over here, in the diagram above, two structures are labelled. Porin and the cell membrane. Outline one function of each structure. So a key word with this is that you are being asked for a function. You're not being asked to describe the structure. You are being asked to outline a function. So what is one acceptable function of the cell membrane? To contain the contents of the cell, to control the movement of molecules into and out of the cell. That's a good go-to one, that second one. And then perhaps if I'm doing a little bit of mind reading, you had a little bit of a panic when you were asked this question. So what is the function of porin? This is not a word, a structure that we are familiar with. Porin wasn't part of the course and it's not part of the key knowledge. So you need to see through the question. The question is not asking you specifics about porin itself. You've been given no other detail about porin other than its structure. So what can you work out from the diagram and with the knowledge that you have from biology? It is a protein. How do I know that it's a protein? I can see the flat arrows that would be the beta pleated sheet structures of a protein. I might also see those coiled structures of alpha helices in a different diagram, but I can definitely identify that this is a protein. Not only that it's a protein, but judging from the diagram, it looks hollow. It is a protein that's embedded in the cell membrane that has a hollow tunnel through it. What can I deduce about the function of porin? That it is a transport protein allowing the movement of larger molecules through the cell membrane. That would be a valid answer. So an answer that convinces an assessor that you have recognized this as being a protein and that you can see that it would be allowing transport. Okay, other kinds of transport across the membranes that we need to be. I think we've talked at length about the first couple of dot points. So other kinds of transport that we need to be familiar with. These are definitely ones that you need to practice recognizing and be able to draw simple diagrams to represent. So for diffusion, have a quick go, if you want to pause the video now, have a quick go at defining each of these before I explain them and see how your memory's gone with the description. And it really comes down to the kinds of molecules that they are transporting. So try to be specific, hit pause, describe these transports across membrane. Okay, so hopefully you have described diffusion, simple diffusion, as being the movement of small non-polar molecules across a semi-permeable membrane, across a plasma membrane, along the concentration gradient. That's an okay way to describe the movement from a high to low region of concentration, or by stating it's moving from a high region of concentration to a low. Osmosis, very similar definition to diffusion, but you have to stipulate that you are talking about free water molecules. So it is the movement of free water molecules from an area of high concentration to low concentration of free water molecules across the plasma membrane. Facilitated diffusion. So again, similar to diffusion, but with the added caveat that we are moving through a protein channel. So why would molecules need to use a protein channel instead of moving directly across the lipids? If they are too large or if they are polar. So only hydrophobic molecules can move directly across the membrane. Hydrophilic molecules being polar, charged, or if they're too large, they would require a transport protein, but still moving along the concentration gradient. Active transport, we're kind of building on it each time. Active transport, similar to facilitated diffusion, we are using a protein channel to move through. However, we are going against the concentration gradient. So this is in an instance where we need a really high concentration of molecule to continue coming into the cell, even when the outside concentration has decreased. So we still need to keep moving, but now it's against the concentration gradient. So actually drawing it in, but this is expending energy. So you would have to, in your definition, have defined that active transport is using energy or using ATP in order to move molecules against the concentration gradient. Endo/exocytosis, you need the word bulk in there. It is bulk movement of large molecules across the cell membrane by use of a vesicle. If we are talking exocytosis, that is removing things from the cell, so exit, things like proteins, larger molecules contained in a secretory vesicle will merge with the cell membrane and release the contents outside of the cell. The opposite to that would be endocytosis, where large molecules are being engulfed by the cell, with phagocytosis being an example of that. And also worth mentioning, so phagocytosis if it is solid material that's being engulfed, penocytosis if it is a liquid that's being engulfed. All right, and just looking at a diagram that's not familiar to you to use to talk about that movement across the membrane and specifically protein synthesis and how it's involved in moving things across the membrane. So take a second to try and digest the diagram that you're looking at here. Hit pause if you need to take it in, because interpreting diagrams is a really big part of being able to prove your knowledge in an exam. So let's see if you can understand what the diagram is showing before I run through it and explain it. Okay, and we're being shown, I would describe, we're being shown probably 3 different processes here. We have some detail, actually not a lot really, we have some detail of what's happening before protein synthesis, in that it's showing the nucleus and the endoplasmic reticulum, and the actually hasn't got any, the mRNA and its role in protein synthesis. But there's a distinction to make. So when VCAR is asking you to describe gene expression, you're kind of drawing a line in the sand before this or during this process, I guess. But here's where the line exists. If you're being asked to describe gene expression, you are being asked to talk about transcription, RNA processing, which will be a later video, and you may have some detail of translation being included there, but it pretty much stops after RNA processing. Once we get into translation, you're now talking about protein synthesis. So if you're being asked to describe the protein synthesis pathway, you're starting from this point. So where the mRNA is being read by the ribosome, and the protein polypeptide has been produced, and this process of excretion starts, or secretion starts. So we'll get into more detail about translation in a later video because that is something you need to build your knowledge on. But once we've got that polypeptide, we're moving through a series of organelles now. So remember, if our protein is destined to stay within the cell, then ribosomes free-floating in cytoplasm will be used to produce that protein. If that protein is destined to leave the cell and go somewhere else, so we might be talking about a gland that's secreting a hormone, then this is the pathway that we'd be following. So if our protein needs to leave the cell, we're using a ribosome that is attached to an endoplasmic reticulum. That protein is, the polypeptide is made, and the primary structure will be made into the endoplasmic reticulum. So we are inside the rough endoplasmic reticulum. As the polypeptide moves through the endoplasmic reticulum, it starts to fold and creates its three-dimensional shape. So you can see the triangles and little stars here represent different proteins that are starting to fold and get their final shape. And again, protein folding is something that we'll talk about in another video as well. Once we've got our protein finished with in the ribosome, in the rough endoplasmic reticulum, part of that membrane then buds off. You can imagine it forming a circle around that protein, and now it is a transport vesicle. That transport vesicle will float to, it's a bit more technical than floating around the cell. The cytoskeleton is actually involved, and if you can see a picture of a vesicle being transported through a cell, it's really cool. But they move to the Golgi apparatus, and that vesicle will merge with the Golgi, and you can imagine it merging, and that protein ends up inside the first part of the Golgi apparatus. Another membrane-bound little organelle will bud off there and form into the next one and the next one. And the way we summarize what's happening in the Golgi to our protein is that it's being further processed and tagged for secretion. So further modification of proteins occurs in the Golgi apparatus. As we exit the Golgi, a new kind of vesicle is formed. You can see it budding away there, and you can imagine a little protein would be encased in that new vesicle, and we now call it a secretory vesicle. And that vesicle will move to the plasma membrane, where it will merge with the membrane, and its contents will be thrown from the cell. And so our protein that was destined to leave the cell has now been pushed out of the cell. You're also being shown in this diagram the process of endocytosis, so where things that are outside of the cell are being engulfed, so a new vesicle is forming around that exterior contents and pulling it into the cell, and it becomes what eventually will be recognized as a lysosome. So this kind of vesicle that's coming in to the cell, we can talk about as being a lysosome. What job, if you can't remember, what job do you think a lysosome would have? Just with what you know about biology and the words that are associated with things. Lysing means to break down. So this is an organelle that has like digestive enzymes that will break down material that can be used. For example, it might be amino acids that are being used to build new proteins in this process here. So it's a nice, neat diagram, and hopefully you could make some sense of it yourself without me having to explain it entirely. And then a quick practice question, hit pause, have a go. Okay, so another unfamiliar diagram, one that you haven't seen before, showing you some things, including iodine, that we're not really sure about, and we've got some organelles that we recognize, a little bit of something going on here. In a VCAR exam, you always have to pay attention to what side, they're tiny arrows, so pay attention to what side that little arrowhead is on, because it will make all the difference to you answering the question. Okay, what are we actually being asked about this diagram that we're unfamiliar with? We're being told that it's a thyroid follicle cell at least. There might have been a bit more question leading into this, but just for answering these ones, all we need is the diagram. Name structure M. So nice easy one straight away, but could you recognize what structure M was? So kind of folded up, it's got dots around its surface and it's in close proximity to the nucleus. Hopefully you recognize that as being rough endoplasmic reticulum. You must write rough endoplasmic reticulum. RER is not an accepted abbreviation. So rough endoplasmic reticulum. Name structure N. And we're going in a bit of a pathway from nucleus to rough endoplasmic reticulum down to structure N, and we know that the thing that comes next in that pathway and what this does resemble is the Golgi body. You cannot just call it Golgi. That is something that I've picked up from examiners' reports in the past. It must be Golgi apparatus or the Golgi body. And name the process occurring at site P. Did you see the tiny arrow pointing inwards? So it's a quick mistake to make to think that this is a vesicle that's merging with the membrane and kicking its contents out, which would make it exocytosis. The tiny arrow is pointing in. So this is actually endocytosis. Hopefully you picked up on that. Okay, and then one more. Bacteria enter through plasma membranes as shown in the figure below. No arrows to watch out for, but you are picking up on the keyword that's saying the bacteria is entering. So it is a bacterium, and hopefully that's made blindingly clear then. Name the process shown in the figure. And pause for you to answer. And hopefully your answer was phagocytosis. I actually think in this year they awarded the mark for endocytosis, but phagocytosis is a better word for it. So that we've got something moving into the cell this time. Actually, it was last time as well. But it is a large solid object, which would be through phagocytosis, which is a form of endocytosis. Okay, so that is the end of this video.